All right, hope everyone's doing well. So we're excited, really excited for this class, CS472 Data Science and AI for COVID-19. So what I'll do today is I'll give you an introduction, sort of overview, and then we'll dive right quite quickly into some of the topics that we want to discuss. So here's like an overview of the class, right? So the class is really meant to be a projects-based class, right? So maybe even more than most other projects classes on campus here. So we really want this one to be, given the nature of the topic, we really want this to be very much project-oriented, right? So I'll talk about this more at the end of today, but we will have a set of 14 suggested projects, right? And then we'll also want to work with you to come up with your own projects and form teams to really work on these different projects. So every week we'll have these lectures, right? Which usually around an hour and a half. So lectures are really meant to provide people with background and tutorials, as well as discussions of some of the latest research. So the class is really meant for students that are coming from sort of data science, engineering, computer science, that's kind of background, right? So I know that people come from quite different backgrounds, and we really want the lectures to first get everybody up to speed about what's the science and public health around COVID-19. And then also to talk about some of the latest research topics. So here we have just sort of plots of the people who are currently enrolled in the class, right? So you can see that actually a lot of the students come from School of Engineering. We also have a fair number of students from the medical school, even a few MDs, and also people from the humanities, sciences, School of Business, Earth Sciences. So it's actually quite a diverse set of students. Right, so it seems like most of the students are actually graduate students, also clinical students, MDs, and also a few more advanced undergraduates and MBA students. Right, so it's really great to have such a diverse set of students. And I think something that we'll really try to encourage when working on these teams and projects is to really try to have diverse teams so that we can all learn from each other. And I'm actually very much looking forward to learning from all of you during this quarter. Okay, so here's the teaching staff, right? So I'm James. I'll be the primary instructor for the class. So my research is really on developing machine learning algorithms for pretty diverse biotech and healthcare applications. You can read about some more of my research on my website. And we also have 4 really fantastic CAs that are helping with the class. So Michael, Arena, Wei-Xing, and Jaime, and they all come from quite different backgrounds, and I think they'll be able to provide also good feedback to the students. So the story of the class is also quite interesting, right? So I think, I don't know for sure, but I think this actually might be the class that came together at Stanford in the shortest amount of time. Certainly, it could be one of the classes that was approved at the very last minute. So basically around March 16th, right? So we had these ideas, okay, can we actually, is this crazy? Can we actually try to do something like this? I was already committed to teach another class, right? So this would be an overload. So we didn't know if we can pull this off. And then 2 days later, I drafted like a couple of paragraphs of email of the syllabus to the CS department. And then, with around 48 hours later, the class was approved, and we had to really scramble to get everything together. Right, so I'm extremely grateful to the course assistants and also to all of our guest speakers for really stepping up, given the short notice to try to get everything together and organize the materials. And we're really excited to make this class a success, especially with the projects. So even though the class is on Zoom, right? So we really want to make it as interactive as possible. So please feel free to ask questions. There are basically 3 ways you can ask questions, right? So I'll try to pause periodically, and I'll also try to get the speakers to pause periodically, so you can just ask questions as you normally would. If you feel more comfortable, you can also just type in questions anytime you want into the chat box, right? One of the CAs will also monitor the chat box and then alert the speakers to the questions in the chat box. You can also, if you want to, there's a raise your hand option on Zoom, so you can also use that if that makes it easier for you to ask questions. But please stop us at any time, ask questions. We want to make this very interactive. And I'll also organize actually some interactive breakout sessions and activities at the end of this class so that we actually can introduce one another, introduce ourselves and make this sort of more informal. Okay, any questions before I actually dive into today's topics? Okay, good. So, let's see. Okay, so the goal for today, so here's the agenda for today, right? So we'll basically have these three parts. So in the first 40 minutes or so, I want to give you an overview of what is the science and public health behind COVID-19, right? So to really get everybody up to speed on the same page. And then the next half, we'll actually have Professor Nigam Shah, who's a professor of medicine. He will talk about what are the Stanford hospital operations related to the pandemic. And then in the end, we'll have actually discussions of the suggested projects as well as brainstormings for small group discussions. Okay, so let's just jump right in. Right, for the overview, I thought it would be important for us to really try to understand these 4 questions. Right, so what is the virus and what does it do? What is the sort of epidemiology around this pandemic? What are the potential treatments, interventions? And what are the broader social economic impacts around this pandemic? Right, so hopefully by the end of 40 minutes, we'll have have some better understandings of these 4 questions. So there are many great resources out there. There are more resources being produced every day. So I particularly want to recommend this resource, the link that I put down here at the bottom, right, from which I actually obtained a lot of some of the materials, which I'll show you in the next few slides. The disclaimer is that many of the things that we discuss here are very much changing by the day, right? Especially some of the science that are being involved, that people are actively working on this, including some folks in my group. So we'll try to discuss what, to our best knowledge, currently what we know is happening, but things could change very rapidly. Okay, so what happened was COVID-19. So in December of 2019, right, so people observed these cases of pneumonia, very severe pneumonia of unknown origin in Wuhan, China, which is one of the largest cities in China. Right, so about a month later, that's when actually the virus that's behind this pandemic was first isolated and identified. And I showed you some of the references here. And then a month after the virus is identified, right, so based on various comparisons that we'll describe, right, so the virus is actually identified to be the severe acute respiratory syndrome, coronavirus 2 or SARS-CoV-2. So that's the name of the virus. And the disease that's caused by this virus, right, that's named COVID-19. So there's one name for the virus and there's one name for the disease, the pandemic caused by the virus. So we'll take a few minutes to actually go through the science behind what is the virus and what does it do. Right, so here's actually an electron microscope image of the virus. Right, so the virus itself are basically these red little spheres, right, red particles. And here these are actually isolated from a patient in the US. And here in this case, the viruses are actually escaping from the host cells, right, from the human cells, which are the blue blobs in the back. Okay, so usually the first thing that scientists try to do when they see a new virus is they want to understand where's the virus coming from, right? So that's similar to if I see a stranger on the street, I'll probably ask her, so which city are you from? What country are you from? So similarly, when we see a new virus, we want to understand, okay, so where's the virus coming from, and what are the relatives of this virus? So it turns out that this virus, right, this behind COVID-19, actually comes from a family of viruses, right? So here we actually have the family tree. So it came from a family of coronavirus. Behind this family of coronavirus, there are actually 4 subfamilies. So you got to serve different clusters of cousins, right? So the one that we're particularly looking at, right, this SARS-CoV-2 comes from this subgroup of the beta coronavirus. Right, so for the purpose here, the exact names of these viruses are not so important. The main thing to note is that we actually have seen relatives of this virus before. Right, so in particular, right, under this family of beta coronavirus, in 2002, there's the first SARS outbreak, right? And that's actually led to about 8,000 confirmed cases, about close to 800 deaths. Right? And then about 10 years later, so in 2012, in the Middle East, there's the MERS coronavirus, which led to about 2,000 cases and also around 800 deaths. Right? So those are also basically the same elements, right, of the same beta coronavirus family. So they're, in some sense, they're siblings of the SARS-CoV-2 that we observe today. In addition to those two, which are more deadly, right, so there are also several other members of this coronavirus family, which leads to these more less fatal, the more common codes. Right? So the names of these viruses are also written down at the bottom. Right, so the first thing that people usually want to do is to, as we said, to understand where's this virus coming from, right? And the way to do that, one of the best way to do that is actually by looking at sort of the phylogenetic tree of the virus, right? So, and also by comparing the sequences of the virus to its siblings, to its, to the neighbors in this tree. Right, so we say that, okay, so the SARS-CoV-2 virus that we face today is from the same family as the original SARS virus from early 2000s. So it turns out that these two viruses actually share around 80% sequence identities. Right, so what that means is that if you actually look at the genome of these two viruses, right, if you just put these two genomes next to each other, right, and just compare them, well, in 80% of the locations, these two genomes actually are the same, right, and in 20% of the locations, these two genomes are different. So in this family tree, right, so you can just view that as sort of the horizontal version of the family tree, right? So the red dots here, right, this cluster are basically the SARS-CoV-2 virus, different strains from different locations that we've isolated. And the top cluster, right, are the original SARS virus. And what is very interesting is that these two clusters are actually, still have a fair amount of differences, about 20% difference. And there are also many of these other viruses that have been identified in animals that are actually more closely related to the current SARS-CoV-2, which led to the hypothesis, right, that this virus actually came from these animals through maybe different intermediary hosts. So in particular, right, so it's been analyzed in this paper, that's the bat virus, right? There's a particular kind of bat virus, which is about 96% similar to the SARS-CoV-2, right? So that's one of the conjectures that actually the virus came from bats. And it was also shown that actually there's particular kinds of anteater, so pangolin, which is potentially even more similar to the SARS-CoV-2, right? So it's still not exactly confirmed the exact path of how the virus actually came to humans, but the one current model is that perhaps it jumped from bat or from pangolin to humans in December last year. Okay, so here's a closer zoomed in version of what the virus looked like. Right, so you should just view that as basically a sphere, a spherical shell. Inside the sphere is the genome of the virus, right? So if your biology is a little bit rusty, so you just view the genome as sort of the instructional program that creates the virus, sort of like the computing, the code behind the virus. And outside of the shell, right, there are a few different types of structures. Probably the most important one are these little spikes that we see here. They're actually called spike proteins, right? So they're spikes that allows the virus to engage and to get inside the human cells. Okay, so the really interesting things are happening when we look at the genome of the virus. Right, so when we actually build these phylogenetic trees, build the family trees, effectively the algorithms behind that is you take these viruses, right? You take the genome, in this case it's the RNA genome, right? You have these letters, you line it up across the genomes of different viruses, and you compare the similarities. And here, basically, we're looking at, on the top row is basically the genome or the protein sequence for this human SARS-CoV-2 virus. And here we have the bats, pangolin, the original SARS virus. And you can see that they're actually very similar across most of the locations. So each column here corresponds to one location essentially in the genome, or one amino acid. The key difference is that there's a particular chunk here of mutations which are quite unique to this new SARS-CoV-2 virus. And these mutations are what currently people believe to be what makes it more easily accessible to different parts of the body. Right? So because of these particular amino acids, right, that's present in this new SARS-CoV-2 virus, that makes it easier to get into the lung and get into the tissue, into the cells in the lung and liver and so on. That's what makes it more transmittable. Okay, so we'll actually have a class later on in the quarter where we'll have Professor Palacios who will give us much more in-depth hands-on tutorial about how do you actually do these kind of phylogenetics analysis, comparing the genomes of the virus to understand how fast is this virus evolving and what are the potential weaknesses of this virus. Great. Any questions so far? Okay, good. So how does the virus work, right? So here's basically a summary of what's happens. Right? So you have the spherical shell, right? That's the SARS-CoV-2 virus. Because of these spikes on the surface, right? That's how it uses the spikes to get inside the human cell, right? So this big sphere here, that's basically the boundaries of a human cell. The virus will get in. It releases its genome, right? It releases its instructional program. The instructional program basically hijacks the human cell's machinery to replicate itself. Right? So the virus is actually too small to have its own machinery. So that's why it actually has to hijack all the machineries within the human cell to replicate itself. Right? So it makes many copies of it. And then the human cell dies, and then different copies of the virus actually releases back out into the environment to infect other cells. Okay. So that's basically the procedure of how the virus progresses. Right? The treatments our people are actively working on are essentially tackling the different aspects of this procedure. Right? So you can imagine that the first step is the virus has to get into the human cell, right? So there are drugs that are designed to try to block the virus from getting inside the human cell. Once the virus actually gets into the human cell, it has to replicate, right? So there are also drugs, therapeutic drugs that people are testing to stop the virus from replicating, right? So maybe some of you have probably heard about like chloroquine, which has been in the news a lot, right? So that's actually one particular targeting a particular part of this pathway of the virus, and there's a few other drugs that people are actively testing in clinical trials of how to target different aspects of the virus. In one of the later lectures, I think in 2 or 3 or 4 weeks, we'll have Professor Russ Altman who will talk about the machine learning algorithms behind these different treatments and how do we use machine learning to improve the treatments. Okay. So we have a couple of questions. So we have a question about, right, so drugs that used to treat HIV and have been used to treat COVID-19, and what do we know about mutation rates of this virus? Right, so these are great questions. So actually it turns out that many of the drugs that are highlighted in this picture here have basically been recycled from previous diseases. Right, so these are not new drugs that we've developed. These are actually drugs that have been recycled, for example, from HIV, from malaria, that are being used to treat these new for these new clinical trials. And the reason for that is that to actually develop a new drug from scratch would actually take much longer time. So that's why people have been mostly, for now, focusing on taking existing drugs that may have even failed for previous diseases, but might work for this new pandemic. The mutation rates. So the second question is, what is the mutation rates of SARS-CoV-2? Right. So that's also a really important question. We'll talk about that exactly that question a lot more in about 5 lectures when we look at the phylogenetic analysis. So so the virus is actually mutating. It's not exactly confirmed yet of whether there's a very strong signal of selection for the virus, right? So the key question is, is the virus actually mutating in a particular way to make it either more or less harmful to humans, right? More or less lethal to humans? Because that would be really important to answer if we want to talk about drugs or vaccines. So that's actually very much an open question, and we'll dive into that a lot more in 5 lectures. Okay, so what happens, so now we understand a little bit about the biology, the science of the virus. So what actually happens to the human, right, when the virus gets in the individual? So basically what happens is that the virus tends to infect these particular types of cells, right? And many of those cells are actually on the surface of the lung. So what happens is that when the virus, right, gets into the cell, that actually triggers the immune system of the human, of the host, to try to fight the virus. Right? So if you're not familiar with the immune system, you view that essentially like the first line of defense. Right? So these are the soldiers that are coming in, right, to really try to fight off like bacteria or foreign agents. So in this case, many of these immune cells will try to come into the lung to try to fight off the infected virus or the infected cells. What actually happens in this case is because you have such a storm of immune soldiers trying to come in to fight off the virus, they actually flood the different compartments of the lung. Right? Because of that flooding, that actually triggers a severe reduction in respiratory function that leads to actually many of the symptoms that you see in severe pneumonia. Right? So that's the result of that is what's called acute respiratory distress syndrome, or ARDS. And that's also why you hear on the news that people actually need ventilators and other equipment because their lungs stop functioning as well due to this flooding from immune cells. And that's why you need to have ventilators to help people to get more oxygen in. Okay, so this covers sort of some of the biology, sort of overview of some of the biology and science. Hopefully that gives you some sense of that. I'll take a pause for some additional questions. We will put all of these slides online on the Canvas, and we'll also put the recordings on Canvas so you can look at the slides and references for more details. Other questions? Okay.
Yeah, it looks like there's a question from Angela and Emily.
Okay, yeah, so Angela's question is, do we know what cell types are more susceptible to infection? So that's a very good question. So yes, there are particular cell types. So the key idea here is that the virus actually has to leverage particular structures on the membrane of the, go back a couple slides, right? So here the virus actually has to interact with particular structures on the membrane of the human cell. So that's one common way for the virus to actually get in. So the particular things that it leverages are these, this ACE2 protein, which is basically a little receptor you find, sort of like an antenna you find on the surface of the human cell. Right, so this receptor actually turns out to be more commonly found in particular types of cells that tends to be on the surface of the lung. That's why the virus is more likely to attack and get into the lung. What are the other cell types that are involved or that are susceptible are something that are still largely under research. Okay, so we have a question from Emily. Right, okay, great. So the question from Emily is, right, so if this, respiratory distress comes from overactivation of the immune system, of the human immune system, would drugs, treatments that sort of suppress the immune system actually help? Right, so that's also a very good idea and very interesting question. So there's quite a lot of active research now on potentially immune suppressant drugs. Right, so those drugs will not really treat the root of the infection, right? So the virus is still in there, but potentially would reduce some of the symptoms, the negative side effects of this immune response of the infection. Right, so yes, so the SARS-CoV-2 virus, it's an RNA virus. Yes, that's right. So what that means is that, just to give people a little bit more of a background, right? So the human genome, right, the instructional program comes from DNA sequence, right? These are the ACTGs. And what typically happens in the human genome is that the ACTGs have to first turned into, into RNA, right, through this transcription process, and then the RNA then is turned into proteins, right? The proteins then do all the hard machineries. So basically what the virus does is that actually it's not DNA, or it comes in as the secondary stage as the RNA, then it can actually be translated into proteins directly. Okay, so the next part we'll talk about is more of now that we understand a little bit about the science, the biology of this virus, so what actually happens to the public health, right? So here's like a flow chart of the progression of this COVID-19, right? And it's based on some of our current best estimates. So the incubation period, right? So that's the time after you get infected, exposed and infected with the virus, and to when you start showing symptoms. So that time typically is around 5 days, right? And after you show symptoms, there could be another week or so where you have relatively mild symptoms. And then based on the individual, some of those can actually show very severe symptoms and for the following weeks, right? And this is where these acute respiratory syndromes, when you need ventilators, could kick in. Now, a lot of the progression, right, is very much dependent on specific individuals. It's very different for different individuals. And part of that difference could be because different individuals have different genetics. Right? Maybe some individuals have certain mutations in their own genomes that makes it less susceptible for the virus. So that's actually also one really active area of research. And there are also some projects that we've been looking at that are involved with understanding what are specific individual differences that make someone more or less at risk for the virus. And in next week's class, we actually have Dr. Kirsch. Dr. Kirsch is actually an emergency medicine physician in New Jersey. So she will actually come in, she's on the front line. She's going to come in and share with us her experience, really seeing what are the symptoms of the virus and how are they dealing with that in the ICU, in the emergency room. Okay, so we have one question from Sharon. So what is on the y-axis of the viral load plot? Right, so this is a schematic here. Oops. This is a schematic here to show for 3 typical kinds of trajectories, right? Over time, what is the viral load in these individuals? Right, so for a typical version, you see the viral load, so the y-axis is the viral load. Right, so you see for this individual, the viral load increases and then goes down over time. Okay, so how does the virus actually get around, right? So that's one question. So here we have like a picture taken from a paper of someone sneezing, right? So when you sneeze, you have these droplets. That's actually quite shown here quite visually, right? You have small droplets, and then you also have some larger droplets. So one common way for the virus to actually get transmitted is at least from individuals to individuals, is actually through these droplets. So one question I want maybe people can think about and put into the chat box is, when you sneeze, how many droplets do you think is actually produced? Just rough order of magnitude. You can put that into the drop box, into the chat box.
And James, while we're waiting for people to respond, there's a question from Faraz earlier.
Okay, let's see. Okay, so we have some pretty good estimates. Some are range from, I think, seems like there's a consensus around 1 million or 10 to the 6. So you see also some things from like 10 to the 5, even larger. Okay, so here are some rough estimates. So when you sneeze, typically you release on the order of 40,000 droplets. right? So most of these are small droplets and a few large ones. So when you cough, right, typically you're releasing around a few thousand droplets. And if we are talking, then it's about a few hundred droplets per minute, right? So these large droplets, right, you see at the bottom here, those are the ones that tends to carry actually a larger amount of the virus, right? So based on the physics, we know that these large droplets would typically maybe travel around like 3 feet or 1 meter, and then they fall to the ground, which is sort of the one of the motivations behind having this sort of 6 feet or 2 meter kind of social distancing between individuals, right? That's basically driven by how far we think these droplets can carry, or at least these large droplets can travel before they fall onto the surface. Now, after the virus actually falls onto the surface, depends on the surface, they can still remain intact and viable, right? So for example, if it stays on plastic or steel, there's some evidence that it could actually still remain intact for a few days. So the symptoms associated with the virus could also be quite diverse. It's not very specific, right? You have fever, cough, fatigue. So the main thing to note here is that these are not super specific symptoms, right? So you could just have allergy and wake up in the morning and have some dry cough and fatigue. So that also makes it somewhat challenging to really diagnose the virus or diagnose the cases. So here's some summary based on a study of the patients in Wuhan of what are the fatality rates and comorbidities associated with the virus. So just to make things more precise, so when I say case fatality rates, what that means is that I look at all the individuals that have been confirmed to be positive for the virus, and we see how many of them actually died, right? So that's among the confirmed cases, how many people died, right? So that's different from how many people overall in the entire population might die from the virus. Comorbidity just means that how many other diseases, right, does this individual have at the same time as they're being infected with the virus. So the case fatality rate, the biggest pattern that people see here is just how much it really increases for the older individuals, right, for the more elderly individuals. And the comorbid conditions are, you can see that's really hypertension, but particularly cardiovascular disease, right? So various heart conditions can really substantially increase the risk of individuals to the virus.
We have one question in the chat box.
Okay, so we have a question from Emily, so Emily asks, do we know how these numbers compare to other countries? So that's a very good question, right? So the numbers could be substantially different. For example, for case fatality rate, some of that depends on the intrinsic biology of the virus in the individuals, right? So across all of the countries, we do see this trend that the more elderly people are more at risk. The specific numbers of how many people actually die also depends on a function on how overloaded is the hospital system in each location, right? So for example, initially in Wuhan, right, so the hospital system was overloaded, but then they very quickly built additional hospitals, right? In places like Italy and Spain, the hospital system is also overloaded, right? So having overloaded hospital systems could also increase the fatality rate.
James, we also have a leftover question from earlier. Do we know why the incubation period for SARS-CoV-2 is longer than other respiratory viruses? And we have a couple new questions as well.
Right. Okay, so the incubation period, I think it's still relatively unknown, right? So I don't think we have a really good idea of what really determines the incubation latent periods, why that's different across the different coronaviruses. Okay, so I'll go a couple more slides and then I'll come back and address these different questions. Okay, so here's just showing you some of the differences between COVID-19 and flu, right? So the numbers, I would say, are, I think, especially for COVID-19, are still somewhat in flux, right? And you should interpret these numbers, as data scientists, you should interpret these numbers with some amount of uncertainty. A big part of the uncertainty is you know that how many people are actually being tested for the virus could really change the outcome, right? So as we said, so the case fatality rate the denominator is actually how many people are confirmed to be positive, right? And how many people are confirmed to be positive really depends on how many people you're testing. Right? So the more people you're testing, the larger that denominator could be, which is why we have a potentially quite a wide range of uncertainty for these estimates. So, right, so we also have, so here are just some plots. I think I pulled this from Wednesday of the total number of confirmed COVID-19 cases in different countries. Right, as you can see these different trajectories. So in 2 weeks, we'll have a discussion with Professor Michelle Berry, and she'll talk about different international policies and policies, public health policies different countries are adopting. And then we can discuss why is it that for certain countries like South Korea and China, you see these plateau shapes, and for other countries, you see longer times take to reach these plateau shapes. Okay, so more locally here in Santa Clara County, right? So this basically those are the data that I actually just grabbed from locally collected here at Stanford. So here are some of the visualizations done by people here at Stanford, right? So we see the number of hospitalizations and number of this is among the people who have been confirmed, right? So confirmed cases. We also see the distribution of the confirmed cases based on their age brackets. So the testing that we're doing in Santa Clara is also ramping up. Of course, as you test more and more, most of the people you test will become, there will be negatives. But we'll talk about next week of how should we account for the different levels of testing when we should build these projection models and prediction for epidemiological predictions. So the last bit I want to mention, and then we'll take a few more questions, is really thinking about the broader economic and social impact around this pandemic. Right, so far we talked about the biology of the virus, right? That's the smallest scale, how the virus affects individuals, that is the human level scale. But the broadest scale is really even going beyond the immediate infections, is what are the economic social impact. This is one plot that I find actually quite striking, right? So the x-axis is just over time. The y-axis measures what is the unemployment insurance claims by week, right, in the US. So this was taken actually over about 50 years. The little gray stripes here actually corresponds to times when there are recognized recessions in the US, right? So the most recent ones from 2008, so you see this stripe, and you see that it's a noticeable corresponding increase in the number of uninsured, in the number of insurance claims, unemployment claims. And what's really just off the chart here is that basically in the last few weeks, right? So you're getting on the orders of many millions of unemployment insurance claims, which is orders of magnitude larger than what was seen previously, even during the previous recessions. Right? Of course, it's still very much uncertain of whether it's going to be a more short-term effect or how long it's going to be the global impact, the economic and social impact of this. But that's something that we have a few projects to think about how to model these global economic impacts. And in particular, we want to look at this through a variety of data sources. Right, so we'll have one lecture from Professor Johannes Eggestad, where we'll actually look at how do we use social media data, like Twitter data, do NLP data mining, how do we use those kind of data to measure different types of sentiments toward the pandemic and sentiments towards also the policies, right? And how do those differ in different locations in the US due to people's different political affiliations and due to their other demographic differences. We'll also have a guest talk from Professor Linnos next week on looking at how do we use survey data to measure the broader impact of the virus, and also how do we measure or even mental health impacts due to the pandemic. Okay, I think I'll take a couple of questions here. maybe why don't the people can just feel free, you can just speak up on Zoom. Maybe that would be a little bit easier, and you can just ask me directly. And Irina, can you check if Nigams is available?
Sure. Let's also maybe start from the backlog. So I've got questions from Addie, Sheridan, Taida, Jing, Blanca, and Josh. So we'll start with Addie.
Yeah, can you just...
I'm here, James, just to answer your question.
Okay.
I'm Addie. I can say my question first. I was curious why, if we know why the top comorbidity is cardiovascular disease rather than some other respiratory disease.
Yeah, that's a good question. So the virus, I mean, a lot of this is still unknown, right? Still being quantified, but the virus actually does impose quite a strong stress onto the heart, right? Which is one reason why, because of all this, also the immune response that actually puts a lot of stress on the heart, which is why we believe that people who already have existing stress or existing conditions on the heart, when they have this additional stress, they're especially at risk.
Thank you.
Okay, who's the next one?
Is there a question from Sheridan?
Hi, thanks for taking my question. I saw something a while back about Germany proposing testing random samples from their population to get more accurate stats. I was wondering if that has begun anywhere, or how Stanford's testing more recently might fit with that.
Yeah, that's a very good question. We'll actually have, I think one of our first projects on serological testing will be quite related to that. So the idea here is that a lot of the testing that are initially done are quite biased, right? In the sense that people who get tested are the people who likely show symptoms or wish to believe that they've been exposed to the virus. So that actually can lead to severe biases when we build these projection models. Of course, ideally, you want to just test everyone, which is what South Korea tried to do. But testing everyone is also just too expensive, especially in the US, given the limitations and capacities. The other approach is to really test people randomly. So currently, the approaches taken here are still not all the way to uniform random testing, right? So even the Stanford, like the approaches taken by researchers at Stanford are more convenient samples, which we'll discuss more next week. But the convenient samples means that we're still testing for people where it's easier to get samples from, right? So we can't just go knock on doors and randomly pick people. If we're likely to get people that are more, who would volunteer to get tested. So that means that there's still biases in the testing results, and we will discuss how do we correct for those biases and account for these biases in our epidemiological mathematical models, but it's certainly still a substantial challenge. So as we mentioned, this is really a projects-oriented class, right? We'll continue to have these Friday lectures and seminars to bring people to bring additional guest speakers in. We have curated 14 projects, right? So we curate these projects so that they have readily available data sets, well-defined questions, and mentors, usually faculties at Stanford, also experts from outside of Stanford who are willing to really work closely with the students. Of course, you're also welcome to come up with your own projects and form your own groups. And Piazza is a very good way for doing that, and we'll help to facilitate, and we can provide resources for your own project as well. So just one sentence about each potential project, right? So similar to how Nigam described it, right? So I've broken down these projects into 5 different types of projects, right? We have projects look at the biology, like look at the viral genetics. And then we also have projects looking at more on the detection side, right? So using their cough sounds or additional testing. We have a few epidemiology projects, which range from looking at mobility data from other sources that are, in addition to the Google data sets, looking at the effects of different intervention policies, government responses. We also have projects that are looking more on the operations side, right? These are looking at supply chains. How do we optimize supply chains for medical devices? How do we also optimize supply chains and distribution of needed treatments? right? And lastly, we have a set of projects looking at really the broader social economic impact, right? That's using, for example, the text data from Twitter, energy data from energy companies, purchasing data from merchants. And a lot of these projects we also want to take slightly a broader view, right? Because as we know, the next few weeks, it's only, in some sense, part of the battle with the pandemic, right? So based on previous experience, we know that there's also potentially a much longer scenario that could last into the fall, potentially into the winter. So we want to also have models for what happens in a few months, right, when the schools come back open again, as well as looking at the mental health impacts of these of the pandemic.